Immunology hypersensitivity. Disorders which are caused by an inappropriately vigorous innate and/or adaptive response to antigens that pose little or no threat are called as hypersensitivities. The term allergy first appeared in the medical literature in 1906, when the pediatrician Clemens von Perkay noted that the response to some antigens resulted in damage to the host, rather than in a protective response. Type 1 hypersensitivity reactions. Also called as allergy and atopy, they are mediated by IgE antibodies that bind to mast cells or basophils and induce vasoactive mediator release. They are mediated by IgE antibodies that bind to mast cells or basophils and induce vasoactive mediator release. These reactions include the most common responses to respiratory allergens, such as pollen and dust mites, and to food allergens, such as peanuts and shellfish. Type 2 hypersensitivity reactions. Also called as antibody-mediated hypersensitivity. It results from the binding of Ig or IgM to the surface of host cells, which are then destroyed by complement or cell-mediated mechanisms. A flat directed against cell surface antigens mediates cell destruction via antibody-dependent cell-mediated cytotoxicity, ADCC, and mainly its target red blood cells, RBCs. For example, this is the fate for transfused red blood cells in transfusions between people differing in ABO blood types. Type 3 hypersensitivity reactions. Also called as immune complex-mediated hypersensitivity. In these type of reaction, antigen-antibody complexes, such as those generated by the injection of foreign serum proteins, deposited on host cells or tissues which activates complement or the release of mediators from granulocytes, often causing inflammatory responses. Typical manifestations include serum sickness, rheumatoid arthritis and system lupus erythematosus. Type 4 hypersensitivity reactions. Also called as delayed type hypersensitivity. Mechanism of action is sensitized T cells release cytokines that activate cytotoxic T cells or macrophages which mediate direct cellular damages. Typical manifestations include contact dermatitis, tubercular lesions and graft rejection.